Today I will be explaining the different point mutations that occur on a single strand of DNA. Here you can see that we took our non-mutated DNA strand and transcribed it into our mRNA coding strand. We then used the trinucleotide bases on the mRNA strand in a codon chart to read which amino acids they code for. In this scenario, they code for methionine, tryptophan, asparagine, alanine, cytosine, stop, proline, and glycine. The first point mutation I'm going to discuss is a missense mutation. This mutation is a mutation in which a single nucleotide is changed so that a trinucleotide base codes for a different amino acid. In this case, we changed a C to a G within the GCT trinucleotide base on the normal strand. This switch is known as a transversion since the pyrimidine cytosine was switched for the purine guanine. The mRNA sequence now reads the base as GGU, which codes for glycine rather than the original alanine, changing the makeup of the whole amino acid sequence and the protein it would code for. This can also be demonstrated through a play on words. If we look at our normal DNA strand as a sentence, in this case, the rad mom got the big red dog, then a missense mutation would read as the rad mom got thy big red dog. This change in one letter led to the sentence being read differently, causing it to have a different meaning. Next, we will look at nonsense mutations. A nonsense mutation occurs when a single nucleotide changes to cause a stop codon to be coded for in the sequence rather than a functional amino acid. In this case, we switched a G for an A in the TGG sequence on the normal DNA strand. This switch is known as a transition since the purine G is switched for another purine A. Ultimately, this causes the sequence to read methionine stop, cutting the sequence short and causing them to code for completely different proteins. Relating this back to our sentences, a nonsense mutation would look like the rad mom period. The early stop causes the sentence to have a completely different meaning that makes no sense in terms of what the actual sentence was supposed to mean. Silent mutations are when a single nucleotide is switched with another, but the trinucleotide base still codes for the same amino acid as the normal strand. In this case, we switched a C with a T in the TGC nucleotide base of the normal strand. This is another example of a transition since the pyrimidine was switched with another pyrimidine. This changed the mRNA sequence from its original UGC to UGU, which both still code for cytosine, allowing for the same amino acid sequence and protein to form. Here we can view our silent mutation as reading the sentence, the rad mom got the big red dog. Mom and mom both have the same meaning when referring to a mother figure based on regional dialect. This causes the sentence to carry the same meaning and translate the same message as the original, even though the spelling was just a little bit different. Finally, we will be looking at a read-through mutation. This mutation occurs when a single base is changed so that a stop codon changes to a functional amino acid. In this case, we switched an A for a G in the TGA trinucleotide base in the normal strand. This is a transition since we went from purine to purine. The stop codon UGA now reads as UGG, which codes for tryptophan. This causes the sequence to continue reading through to the next amino acid proline rather than stopping after cytosine. In our sentence, a read-through would look something like the rad mom got the big red dog and. Rather than stopping after dog, the sentence would continue reading on to another functional word such as and.